My name is Paige Sedler, and I'll be talking about our gut microbiome experiment. So a microbiome is the different types and proportions of microorganisms in a specific location. And for our research purposes, we examine the gut microbiome. The gut microbiome has been shown to correlate with a considerable number of various physiological conditions. The first physiological condition that we found had relations with the gut microbiome was Alzheimer's disease. Alzheimer's disease is the most common cause of dementia and has been associated with bacterial infections and inflammation. The latest studies examining the intestinal microbiota of Alzheimer's patients shows that their microbiome differs from non-dementia having patients. Via stool samples, it was determined that elders with Alzheimer's induce lower P glycoprotein expression in vitro than samples from elders without dementia. We also paired function with machine learning approaches to identify bacterial species differentiating in the microbiota of AD elders from elders without dementia. The relationship between microbiome and Alzheimer's is correlative because studies have shown that a person's gut microbiome with Alzheimer's is different than a person without Alzheimer's. And the second and final physiological condition that we explored was Parkinson's disease. Parkinson's disease is one of the most common neurodegenerative disorders. The latest study showcased changes in the gut microbiota in patients with Parkinson's, which served as both biomarker of Parkinson's or trigger-inducing folding of an A synuclein, which is a component of Lewy bodies that launch neurodegeneration into effect. The relationship between gut microbiome and Parkinson's was deemed correlative as well, because the study showed that the gut microbiota in Parkinson's patients is characterized by a decrease in taxonomic diversity and significant differences in the 9 genera and 15 species of microorganisms. In conclusion, it was our thesis, our hypothesis that the 2 dogs that we used for our experiment would have different gut microbiomes due to gender, despite otherwise being similar in age, health, weight, and ownership. And the sources for that are here. And then as far as materials go, first of all, the animal samples that we used were sample 1157, which is a Chihuahua male, 4 years old, in good health and neutered. And then the second sample was a Chihuahua, which is the sister to sample 1157, a female, 4 years old, spayed, 6 pounds, and also in good health. The third sample we used was 0310, which was a miniature horse. It was 10 years and 6 months old, not neutered, very good health. And then the fourth sample was 0885, which is a horse, male, 14 years old, 1,100 pounds, and also in very good health. And then a brief introduction to the protocol. First, isolate the DNA from fecal samples using a PCR kit. And second is to quantify and normalize the DNA. And third is to analyze the quantity of specific bacterial groups using PCR reaction, polymerase chain reaction, with group-specific primers. And as far as the isolation of the DNA, commercial kit used was the QIAGEN qPCR kit, which is a laboratory technique of molecular biology based on the polymerase chain reaction. It's commonly used to monitor the amplification of DNA molecules during the PCR, and most commonly associated with the endpoint method used to amplify DNA or RNA fragments. And it can detect different nucleic acid sequences in a given sample. And as far as quantifying and normalizing the DNA, the instrument used was the spectrophometer. This instrument is able to determine the average concentrations of nucleic acids in DNA that's present in the sample. And how it works, it's a digitalized computer, and it takes in light, it breaks down into spectral components, and then it's able to digitalize a signal at a certain wavelength. And once the process has been done and calculated, the quantification will appear on the computer screen as just a unitless number, which is absorption. And as far as the PCR setup, Primers that were used were the bacteriodutes and the firmicutes. And these primers are important because they're specific in silico and against any known dog. And Cyber Green is a fluorescent dye that binds double-stranded DNA molecules between DNA bases. It usually is used in a qPCR because dye can be measured at the pH of each amplification cycle to determine how much DNA has been found. And on the results table, this is the UV spectrophometer DNA quantification. So for sample 1155, which was the dog, the male dog, absorption at 260 nanometers was 1.17, and the DNA concentration was 58.9, and the 2060 to 2080 ratio was 1.97. And I'll get into explaining what all these mean in the discussion. For sample 1157, which is the female dog, the A260 nanometer reading, which is absorbance at 260 nanometers, was 0.328. The DNA concentration, 16.4, and the 2060 to 2080 ratio, 1.53. For sample 310, which is the first miniature horse, absorption at 260 nanometers, 0.772, DNA concentration, 38.6, and the ratio 2.15. 0885, which is the second miniature horse, 0.188 absorption at 260 nanometers, DNA concentration of 9.4, and then the 2060 to 2080 ratio of 3.27. And for our first qPCR, we had positive, negative samples as well as our regular samples. So for the U bacteria primer, the CQ value for the positive control was 14.2, and the antilog was 5.3 times 10 to the negative fifth. And for the negative control, the U bacteria CQ value 29.5, and the antilog 1.32 times 10 to the negative 9. For sample 885, CQ value for U bacteria 17.33, and antilog of 6.1 times 10 to the negative 6. Sample 310, second dog, 16.77 CQ value for U bacteria, and 1.4 times 10 to the negative fifth antilog value. Sample 1157, CQ value for U bacteria of 25.07, and antilog value of 2.8 times 10 to the negative 7. And for our final horse sample, 1155, U bacteria CQ value of 19.92, and antilog value of 1 times 10 to the negative 6. And then for the fungi primer, the negative control showed a CQ value of 36 and an antilog of 1.5 times 10 to the negative 10. And still going with the fungi primer, for the sample 885, CQ value of 39.22 and an antilog of 1.5 times 10 to the negative 12. Sample 310 CQ value for fungi, 32.53, and an antilog value of 1.6 times 10 to the negative 11. For sample 1157, CQ value for fungi of 30.45, and an antilog value of 6.8 times 10 to the negative 9. And then for sample 1155, fungi CQ value of 37.19, and antilog value of 6.4 times 10 to the negative 11. And going on to qPCR number 2, for the positive control, the phylo 1, which is the bacteriodutes, CQ value 32.31, antilog 1.88 times 10 to the negative 10. Negative control group for phylo 1, 0 for the CQ value and 0 for antilog value. For sample 885, 37.27 for CQ and 6.05 times 10 to the negative 12 for antilog. For sample 310, 19.09 for the CQ and 1.79 times 10 to the negative 6 for the antilog. For sample 1157, 26.42 for the CQ and 1.11 times 10 to the negative 8 for the antilog. And for sample 1155, 19.96 for the CQ and 9.81 times 10 to the negative 7 for the antilog. And then moving on to phyla 2, which was the firmicutes, the CQ value for the positive was 23.35, antilog 9.38 times 10 to the negative 8. For the negative control, CQ 30.11, antilog 8.66 times 10 to the negative 10. And then for sample 885, CQ 18.03, antilog 3.73 times 10 to the negative 6. For sample 310, 17.68 for CQ, 4.9 times 10 to the negative 6 for antilog. And then for 1157, 26.63 for the CQ, 9.64 times 10 to the negative 9 for the antilog. And for the last sample, 1155, 23.12 CQ, and 1.1 times 10 to the negative 7 antilog. And then there's also the phyla 1 to phyla 2 ratio. And for the positive control, that was 2.0 times 10 to the negative 3. For the negative was 0. For 885, 1.62 times 10 to the negative 6. For 310, 3.65 times 10 to the negative 1. For 1157, 1.15. And for 1155, 8.96. And then when extracting DNA from the scat, extraction was successful in terms of the quantity of the DNA. This means that the DNA concentration was abundant enough to be able to read using a specialized spectrophometer at 260 nanometers. The absorption at 260 nanometers for sample 115 was as read. And then the samples were also sufficient in terms of quality, meaning that the DNA samples were not contaminated with protein. That's based on the 2060-2080 nanometer ratio. Pure DNA has a 2060 to 2080 ratio of approximately 1.5 to 2.2. And then the ratios below this indicate possible protein contaminations. And all of the ratios for the samples were within that range. And then the results of the first qPCR for the U bacteria positive control was, as I said, 14.2, meaning that the target DNA was very abundant, which is expected in positive controls. And the first CQ value for the fungi positive control was unable to be read. This can indicate an issue with instruments as well as and/or technique. The first CQ value for the U bacteria negative control was 29.5, meaning that the target was basically absent, which is expected in negative controls. And then the first CQ value for the fungi negative control was 36, which also indicates an absent target. And then the results for the second qPCR for the Bacteroidetes negative control was unable to be read, again indicating an issue with technique or instruments. The second CQ value for Firmicutes negative control was 30.11, which again indicates an absent target, which is expected. And then for the results of the third qPCR for the Bacteroidetes negative control was a CQ value of 21.46, indicating a rare target. Ideally, the CQ value for a negative control should be greater than 25. However, errors with technique, instrument, cross-contamination can all affect these values. The third CQ value for the Firmicutes negative control was 25.12, again indicating an absent target. And there were differences detected within the gut microbiome between all animals that compared. The dogs compared sample 1155 and 1157 were both 4-year-old Chihuahuas from the same household, sample 1155 being the female and 1157 being the male. The second qPCR shows that sample 1155 has a bacteria to Firmicutes ratio over 7 times greater than the ratio for sample 1157, 8.96 and 1.15 respectively. And the two horses compared, samples 0885 and 310, also showed differences, with sample 310 having a bacteria to Firmicutes ratio of 0.365, which is significantly higher than sample 0885, which had a ratio of 0.0000162. And then the data collected provided insight to our question, will gender have an effect on gut microbiome? The data supports the hypothesis that despite the two dogs being the same breed, age, and living in the same household, their gut microbiomes will differ. A future experiment to think about would be finding out if the differences in the gut microbiomes were actually due to gender or if they were due to some different underlying cause or group of causes.